We have two speakers today in our seminar. So first, we're going to... Thank you, Linda. First, we are going to start out with Dr. Paul Glaum, who's a postdoctoral scholar in the Department of Environmental Science and Policy at UC Davis, working with for non-bovino pollination biology and theoretical ecology, and his ecology and flower special pollinator net. So Paul is after that, we'll have questions and the next speaker. All right, so over to you.
Great, thank you. I will start my screen share. Okay, come on. Is everyone seeing the screen okay? Fantastic. All right, thank you for having me. As was mentioned, I'm over at, or I guess I'm employed at UC Davis, but I actually got my degree here. At this point, it's been long enough that if we were in person, I think I'd be meeting many of you for the first time. But regardless, I'm happy to be back, even if only digitally. Today, I want to talk to you about some of the work that was actually started here at U of M and made the trip with me to Davis. And I wanted to quickly mention that this work was done with obviously a number of people. Obviously, my advisor found the ovBBx was also done with Jonathan Morris, or Jono, as you may know him, over at SEAS, and our pollen expert extraordinaire, Thomas Wood, who's a worldly British researcher who actually met at MSU, who's now living and working in Belgium. Okay, so this is not a talk about my collaborators, it's a talk about diet breath. Why are we talking about diet breath, particularly in pollinators? Well, the diet breath is an kind of important consideration. We think it sits at like the nexus of a lot of kind of central questions in ecology and evolutionary biology, ecological references, inferences along the lines of the fact that diet breath determines competitive interactions. It can predict species persistence in the face of disturbances. It influences dynamics both at the population and community level. Evolutionarily, it can influence diversification and lineage differences. It can determine the ability to migrate or disperse, influence gene flow. So yeah, we think it touches a lot, and we think that understanding some of the drivers of diet breath or diet specialization are going to be important to kind of getting some inference on a lot of central questions in biology in particular. So we asked first, if we're thinking about this, how broad are diets? And we can look at the literature and see what's out there right now. And we can see that some recent work finds that frequently, at least in insect herbivores, diet breath tends to be somewhat specialized, at least at the family level. This is a big study from 2015, found a high degree of specialization at the family level. And this was largely due to insects needing to become acclimatized or defense to the plant's chemical defenses. Here you can see these bar graphs show the family level plant diet breath of different insect herbivores, orange showing lepidopteran and green showing all herbivores in general, all insect herbivores in general, at least that were tested. So we see a kind of at the family level there. But we're interested in pollinators, so we kind of look at the available to us, and we can see that pollinators, particularly bees, show a real mix of generalists and specialists. Only about 30% are going to be specialists in a lot of temperate areas, and there's kind of a range from specialists to generalists that you often see in this this nested network structure that a lot of pollination networks have. So that kind of begs the question then, if we have this mix of generalists and specialists, often within the same area, what drives diet breath in pollinators or diet specialization, particularly in pollinators, is what we're interested in here. So we are obviously not the first ecologists or biologists to think about this. There's been a lot of work done on this generalist specialist question. We want to kind of start with a historical context, kind of what drove us to get here in the first place. starting with a neat paper from back in the 60s from MacArthur and Bianca that looked at a bunch of different factors that influence the push toward specialization for hunting animals. And then compare that to more recent work in the 90s from Wasser et al. That looked, they kind of had similar points, but kind of took them in a different direction to look for what favors generalization in pollinators. Now, this is a big table of text. We don't need to go over all of it. Just kind of wanted to show you this for historical context. Is we're going to focus on this first part here that looks at the common factor of the density of resources measured relative to the foraging range or foraging period of the animal in question. In our case, pollinators. So, seeing as how we're kind of talking about space on one hand, like a temporal aspect on the other, we should get into detail of what we mean here by density. Compare these two ideas, just to kind of make the transition. So the first one, physical geographic density. This is something everyone's pretty aware of. It's relatively intuitive. If you have two separate resources, kind of here in this toy example, plant 1 and plant 2, you can see that plant 2 is 4 times more abundant than plant 1. A pollinator, as long as it has the flight ability to reach all these resources in this area, it could potentially specialize on plant 2 because it has that ability to interact with those options. But we may be stretching the definition of density here a little bit, but just for kind of our framing purposes, we are considering that there are other aspects of density to consider, and it's not just geographic, but it's also temporal. What is the temporal resource density of resources across time? So if we have a growing season where we have the start of the year, at the end of the year, we know that especially for flowering resources, they aren't constantly available. There's times when they are available and times when they aren't. So that's going to influence what a pollinator can go after. If we have this kind of chopped up amount of options for resources across time, what does that do to the diet breadth of the pollinator? So we argue effect of better diet is a function of what we're calling the temporal overlap among different flowering resources. That's kind of our connection to the density, how much overlap of resources there are in a particular moment in time, and how that interacts with the foraging phenology or the flight period of the pollinator. Okay, so let's get into the detail of that. What exactly do we mean? So here's kind of the first part of our diagram that describes our hypothesis. We have the bee and the bee foraging time or flight period shown in orange, and different plant flowering time shown in green here on this kind of this axis from spring to fall, or just any kind of growing season. We can see that if we have a long flight period bee, kind of akin to a long-lived foraging animal, we see that if you have a low overlap environment shown there on the top from spring to fall, or what some people have called the literature a highly seasonal environment, where the different options do not overlap very much. And then you compare that to a high overlap environment or low seasonal environment where there's a lot of overlap among the different flowering resources. If you look at the kind of temporal for any one bee in particular, there isn't actually a lot of change for this long flying bee. Whether it's in the lower overlap or high overlap environment, there's not much change. So what we see is temporal resource overlap and specialization don't necessarily explain each other in this long flight period bee. Okay, so I know I started our hypothesis with a non-effect. So let's get into the part that's we're actually we expect to see something. And let's look at the short flight period bee. So a short flight period bee has a obviously smaller window of time to search for resources. So the amount of temporal resource overlap is then going to have a differential effect on what's even available to search for in the first place. So in our kind of diagram example here now, we have a short flight period bee, like a bee in the Andrena genus. And the low overlap environment, in this case, it only has one option in general for floral resources. But then if you look at that same kind of temporal location, but in a high overlap environment where there's a lot of different resources overlapping one with the other, we can see that the number of options goes up, in this case to 3. Obviously, if we kind of shift where that entry in a B flight period is in our diagram here, we could maybe get like 2 options in 1 compared to 3. But this show here is that in general, you can see that there's going to be a difference between the low overlap availability and the high overlap availability for the short flight period. So this, in the long and short of it, is our. Didn't mean that to be a pun, but it came out. The long and short of it is that the longer flight period B is not going to see much of an effect of temporal resource overlap on its diet breadth, but for the shorter flight period B, we are going to see a reduction in specialization as temporal resource overlap increases. Okay, so that's our hypothesis. How do we study it? Well, we took a dual approach where we have theory and empirical, not necessarily working together. We're not comparing, or I guess I should say here, like they do in epidemiology, but we're looking for qualitative similarities between our results. And our theoretical approach takes a plant pollinator network model that has adaptive foraging in the pollinators that allows them to choose what resource they want to focus on, given what gives them the most benefit. And we're going to modify that model with phenological expression for B flight period and flowering time. And then the empirical side of things, our pollen expert extraordinaire collaborator has collected a large intercontinental data set of pollen from short and long flight period bees in high and low overlap environments. So let's talk about the modeling framework quickly. So plant pollinator models are often done such that the interactions happen on a network, and that's what we're doing here as well. And again, just to reiterate, the pollinators on this network have adaptive foraging potential, so they can choose where they search for the resources. But what's different here in typical models of this sort, the interaction of, or I guess I should say, the potential for this interaction to occur, any particular interaction that is linked in the network is always on. It's always active. No matter where you are in the simulation. But because we're interested in phenology, we want to be able to change that. And the way we do that is by modifying the flight period or foraging time, and then the flowering time of different flowers, such that if there is a potential interaction for a bee and a flowering plant, it needs to be during the kind of zenith of their overlap of flight period and flowering time. And that's when the interaction is the highest, or has the highest potential to happen. Otherwise, it is not possible to occur. So that's what we did. We modified in our network to have a phenological expression of flight period, and we modeled every single plant in the network to have a phenological expression of flowering time. Kind of toy example here in the slide of what it looks like when you blow it up to a full network is you can have different links in the network active at different times. So if you have the pollinators here shown in orange, the reason it says A is because it's animal pollinator, if you're curious, because we can have P for pollinator and P for plant. If we have A1 for animal pollinator 1 and we look at T1 and then compare it to the network, we can see that it is potentially interacting with plant 1 and plant 2, but not plant 3. And then we go to T2 in blue, and we see that the animal pollinator 1 is no longer active, so those links are no longer active, and the links now interacting between A2 and P2 and P3 are now active. Okay, just to kind of quickly describe the setup of these theoretical experiments and the model specs, we have a large number of different plants and combinations of plants. That should say pollinator and plant phenology parameters, creating a range of temporal resource overlap, so we can kind of go from low resource overlap to high resource overlap on a continuous basis. And what we did with our plant pollinator networks is we started them out fully connected. such that every pollinator had the potential to go to any plant as long as that plant was active at the same time the pollinator was, because we didn't want to a priori restrict the ability for any one pollinator to act with any one plant, because that would change our readout of diet specialization. And if you're curious exactly what that looks like to go from non-connected to fully connected, we have a slide at the back end of this talk. I can show you to show what it looks like. But basically, you can take a empirical network and just kind of fill in all the missing spots to say, in our network runs, if the time overlap is possible, then we allow the bees to potentially make that interaction. But yeah, over the course of our different iterations, we ended up with a large number of results. And just to kind of describe exactly what the modeling gave us, at the end of every single simulation, we can look at the flight period of the bee to see if it was long or short or somewhere in between. We can then get an output of the amount of temporal resource overlap. If you have questions about exactly how we quantified that, we can talk about that later too if there's questions. So we have like how long the bee was flying, how much resource overlap there is. We have a readout of where it spent its time foraging. So we can get an idea of how broad its diet was in our model. Okay, on the empirical side of things, we determined plant-pollen interactions through pollen loads on wild bees, such that Thomas here literally small amounts of pollen on the scopa or the kind of the pollen basket of these bees, and painstakingly went through them and identified them to see what was there. This was done in 2 locations, again here in the United States and the United Kingdom, and with 2 bee genera, Laís and Andrina. So why do we make these choices? Well, for bees, Laís is a long flight period bee, which fits our purposes, and Andrina is a short flight period bee. So we have this nice dichotomy between a long and short flight period, which we need to test our hypothesis. Also working within genera can control for the effects of phylogeny, evolutionary history, or physiological differences that can influence where a bee will forage. There are 2 of the larger genera in the world, which is great because we have a number of species to look at. And both genera as a whole tend to, or at least in the regions where we're studying, favor shallow, open, symmetrical flowers, just we can be kind of comparing like with like. And if you're interested, just against some extra evidence of these actually having long and short flight periods, with our data, we can look at the dates of collection of Andrina and Laís and see that the Andrina bee species did have a smaller window of time which they were collected compared to the Laís in both locations. Speaking of locations, we have Michigan and the UK. Michigan is a highly seasonal environment, such that flowering overlap is not very high. It's quite low, actually. Flowers tend to come in like different peaks across the year, compared to the UK, where it's a more temperate, less seasonal environment where there's longer flight periods, so there's a lot more overlap. Another nice thing about these 2 locations is you have 2 hollow Arctic locations with similar plant families. Each location had roughly 40-ish plant families, and there's about 26 overlapping, so that's helpful. So we have kind of a similar composition availability of the rewards. And just to show a quantitative measure of overlap in these 2 locations, we did kind of a deep dive in the literature to look at any kind of phenology research we could find, which was really easy in the UK because they have this amazing database called Ecoflora. And in the Michigan, or I should say in the United States in general, but in Michigan in particular, it was a hodgepodge of data put together through various methods. And we can see that if you look at the pairwise overlap of any one species versus another in each location, we can see that Michigan is substantially lower than the UK. So the UK has a much higher amount of resource overlap between flowering species. Okay, so how did this pollen identification work? Well, Tom would be the better one to ask, but since he isn't here, and say that it was not something I'd want to do. It was removed and sampled with light microscopy over 120 bee species, 2,561 individual pollen loads, lots and lots and lots of individual pollen grains, gave us pollen ID to plant genus of 166 unique plant genera. And finally, how did we measure bee specialization? Well, we used a number of metrics, but to describe them would take a little bit of time for a relatively short talk. So I wanted to kind of stick to the most probably, at least what I hope is the most widely used metric, such that people kind of have an understanding of what we're talking about. That is the coefficient of variation. And you can see how coefficient of variation kind of will go up, the more of an outlier you have. Compared to the more how even the foraging effort is for the bee. So in this case, high levels of CV or our degree of specialization are going to mean high amount and low are going to mean low specialization. Okay, results. Theoretically, this is an example. Our results matched our predictions pretty strongly. Here we have an example in figure A of, one thing I should mention quickly is that we took our specialization metrics at the absolute end of the simulation and averaged across time to make sure that the end of the simulation wasn't an outlier and that we could compare those two and at the next have an average. But in general, we took the diet breath at the end and the average across the end over the last couple thousand time steps. In this case, measuring at the end here for this particular example of the results, we can see that the short flight period B, in this case 20 time steps, has its degree of specialization explained pretty strongly by the amount of resource overlap, in this case measured as the average per individual plant species. And we compare that to the longer flying bee, and we can see that the explanatory of resource overlap really drops significantly, and it explains a lot less, especially in comparison to the short flight period B. And it's kind of a nice representative sample of the effect of resource overlap explaining the degree of specialization for different flight period Bs. We can see that generally, as the flight span of the bees in our model goes up, the kind of the explanatory power of total overlap. So that's great. That works for us, and it's this nice kind of continuous example. We can see that our mechanism is actually something we can kind of ramp up or ramp down. But then now we have to compare it to our empirical results. And luckily, our empirical results happen to also match our predictions. Just recall that Michigan is our low overlap environment, and the UK is our high overlap environment. If we look at the lacer glossum, being our long flight period B, we can see that there is no real effect size, or there's no result to report. It's not significant. But then when we look at Andrina or short, we can see that the specialization in the high overlap environment of the UK is significantly less than the specialization in our low overlap environment. So that's great. Seems to check out. So to summarize, the degree of overlap range of diet breath and pollinators, shorter flight periods or the foraging time, such that high degrees of temporary resource overlap facilitate foraging, and low degrees of temporary resource overlap kind of force a more specialized foraging. They have less options, so they tend to be more specialized. And then for longer or longer flight periods in pollinators, the effect of temporary resource overlap on diet, and our results were qualitatively mechanistic simulations and our empirical data sets. So that worked out. Future directions. Our minds went relatively quickly to being able to study what's happening with phenology in the real world. The climate is changing. The phenology of things is also changing, and there's a lot of questions to ask on what that's going to do to existing plant pollinator networks, how resilient they might be, or how they might change in general. And of climate change, that brings me to my final point here, which is that the University of Michigan opportunity to take some of its financial that are burning fossil fuels, leading to climate change, leading to these phenological changes that we're seeing and studying, which maybe means less work for me, but hey, that's okay. Speaking for myself, I guess, and the bees, I think they should do it. I'm just wanting, just want to get out what the bees have been telling me. I've ever spoken has said this. So yeah, passing it along. All right, thanks and acknowledgements, obviously to my collaborators, couldn't have done it without them. To our computing resource funding. If you have any questions, if there's any references, I'd be happy to take them. Thank you.
Thank you, Paul. And so Katrina, our host, is having a little bit problem with internet connection. So I think I will take over to host the seminar from now. So now it's a question time. So anyone have any questions? You can either type that in the chat or you can unmute yourself and speak up. Okay, a question from Kyla. What results would you anticipate for tropical environments?
Tropical, so if you look at bee specialization in species in tropical environments, they tend to be a bit more, well, I guess there's kind of a range, because you definitely have your euglossine bees, which can specialize relatively heavily, but you also have some very generalist bees. So there's a lot of resource overlap in lots of places in the tropics, such that we don't really think you'd be able to use it very well as a predictor for especially some of the longer lived bees. And because a lot of the bees tend to be longer lived there, it's interesting to wonder how much of an effect it'll have. But in the tropics, when you have these longer lived bees, and obviously along well, a never-ending growing season, depending where in the tropics you are, you might actually see more of an effect of things like physiological differences in plants and bees. Yeah.
Okay, so another question from Catherine. Are pollinator flight visitations affected by pollinator interactions? I'm wondering about areas where the honeybee is common and its impact on pollinator flight times of native bees.
So for the longest time, to my understanding, there's kind of an open question of whether we can find good evidence of honeybee colonies competitively impacting wild bee species. And it took a little while to get good data on this, but I think over the last couple of years, it's becoming increasingly well known that there's at least some effect, and it can differ depending where you are. So there's definitely a competitive effect. What it may do to any kind of flight period is, I mean, I don't think it would change the flight period so much of any bee it's competing with, but it's interesting to consider, because one of our next steps in our model too is to start running these simulations such that we have longer flight period bees competing with short flight period bees, and see if we can get similar results. Preliminary results were promising in terms of matching our hypotheses, but it'd be interesting to put a honeybee type bee into the model such that it has such a long flight period, especially in these temperate areas, compared to a lot of the solitary short flight period bees, and see what that does. I mean, I would imagine you would see a competitive effect, and it would be interesting in the model to balance out the competitive effect that could potentially cause extinction of competitive or competing pollinators and finding some weird parameter space such that you can keep everyone alive, but see what the honeybees are doing to the foraging patterns of the shorter flight period bees. Especially for honeybees, because they're so generalist. So they kind of take from everywhere.
Okay. Because of the time limit, I think we need to stop here and go to our next talk. So let's, if we still have time in the end, we can go back to other questions. But now let's thank Paul again for the great talk. And our next talk would be given by Laís Petri. So Laís is a PhD candidate in EEB lab at SEAS. She studies invasive plant species to understand how invaded plant community assemble to inform management. Her talk today is titled Using a Community Assembly Framework to Decrease Vulnerability to Biological Invasions in Temperate Forest. So let's welcome Laís.
Hi, thank you. Let me share my screen. Okay, are you seeing it? Yep. Okay, thank you. Good afternoon. Thanks for having me here today. It's really nice to be here and show my work. I will present the research I'm exploring during my PhD at SEAS. The main idea here is to study why and how we can use community assembly framework to make forests more resistant to invasive plants. So ecologists have long been interested in understanding the mechanisms behind plant community assembly. But recently, this interest has renewed strength within the context of plant invasions. Community assembly is especially relevant in the current environment where the movement of species is a worldwide phenomenon. As new species are introduced into an area, some may become invasive and then harm the native community. So current research approaches to biological invasion emphasize on the traits of the non-native species, the context under which they become invasive species, their interactions with native species in pairwise comparisons with native species, and they also focus on trying to estimate the risk of particular invasive species could pose on the native communities. And despite these efforts, there is still great uncertainty in predicting which non-native species will become invasive. And this uncertainty hampers a lot target management of vulnerable communities. So I propose to switch the research focus from the invasive species to the affected community by investigating the potential role of two mechanisms driving community assembly during invasion, which are empty niches and priority effects. And my main questions are, so the first one is what drives forest resistance to invasion? And then what are the mechanisms driving community assembly during forest invasion? And then which circumstances those mechanisms operate? And can we use this knowledge to actually inform management actions on the ground? So for long-term persistence of healthy forests, native plant species recruitment is critical. However, when invasive species, they occupy forests understories, they threaten the establishment and survival of the native communities. And due to an intensification of the anthropogenic disturbances and the constant movement of plants across the globe, forests' vulnerability to plant invasion has increased in the past decades. But still there are instances where the forest communities can resist invasion, and those resistances, they happen when the ecosystem imposes some barriers to invasive species in order to slow down establishment growth, increase in abundance, survival, or reproduction. But and there are several mechanisms that would be underlying forest resistance to plant invasions, And I will personally focus in two of them that I will further discuss observational and experimental approaches to try to understand them. The first mechanism is empty niches. So plant communities, they maximize the use of limiting resources. But there are instances that not all of the resources that are available, they are being fully explored, exploited, and utilized by the native community. And there are two main reasons why those resources may not be completely used. The first one is that shared evolutionary history in resident communities may constrain co-occurring species to maximize resource use via negative feedback in fitness and in performance. And I will give an example for temperate forests, which is my study system. So light access is constrained by the canopy in temperate forests. For native species, leafing out early, but not too early before the canopy closes to exploit this light availability has likely evolved as a strategy to avoid the damage from late frost events. Even though there might be optimal conditions to photosynthesis, before they actually leaf out, those native species in the understory have evolved to avoid this window of opportunity due to some risk of frost. Then there is this available niche of light availability that with the frequency and the severity of extreme cold events, they are predicted to decrease due to global warming. And this strategy may no longer be so advantageous to those native species in the understory, especially to the ones that are least phenologically responsive to those changes in the warming conditions. But some invasive species are already predisposed to take advantage of that extended growing season. And many invasive shrubs leaf out earlier and postpone their leaf senescence compared to other native understory species. And these invasive species use the higher light levels before the canopy leaf out and after the canopy senesce that are not being used to the same extent by the native community. And then they are exploiting this empty niche. The second reason for the presence of empty niche is environmental heterogeneity. So it promotes variability in resources, in time and space, variability that native species and native communities are adapted to. For example, native disturbances such as the windrow that I show here in the picture, they decrease plant biomass and temporarily reduce the use of the available resources. So the native communities are immersed in this shifting landscape where the resources supplies vary, but the variation that they are evolved to respond to. But when we have novel temporal or spatial increases in resource inputs or novel decreases in resources use, they all might result in an incomplete resource exploitation by the resident community, leaving behind some untapped resources. So in anthropogenic landscapes, resource availability shifts may not match to those that the native communities are adapted. And if native species cannot utilize these untapped resources, invasive species might be able to. And by using those empty niches, they might become invasive. So in order to investigate empty niches, I plan to use species functional traits together with estimating resource availability. And because the trait analysis can be a useful approach to evaluate the relationship of co-occurring native and invasive species in the context of the local abiotic environment. So here I present some hypothetical scenarios in how this link can happen. So let's consider here this graph in which we have species abundance in the y-axis and above ground traits in the x-axis. And they are moving, the above ground trait values, they are moving from the far left from slow or conservative species to fast traits acquisitive species. And I have here my native species in the gray dots. So in disturbed environments, they display slow to fast traits. But for invasive species, I hypothesized several alternative behaviors. So here in this scenario, invasive species represented in the pink dots, they would succeed due to mechanisms other than distinct resource use traits. For example, higher competitive advantage by having higher abundance and same or similar trait values than the native species. And this another alternative where the invasive species are represented by the blue dots, invasive species possess similar above ground traits and abundance compared to the natives. And their success is due then to other mechanisms. So for example, propagate pressure. And my last scenario here, where invasive species are in orange, the invasive species would succeed by holding faster traits and thus exploring niches that native species are unable to. They have a different set of traits to the far end, the fast traits, and have higher abundance compared to the native species. So let's go to the second mechanism that I talked before that I want to explore, which is priority effects. So priority effects is a mechanism that might occur when community assembly is reset. So for example, in my picture about the wind throw again, there is a decrease in competition after this disturbance event. Plant biomass was removed, some limiting resources that were used by those previous plants are now available, including space. And with that, especially native species with acquisitive traits that establish first via seed dispersal or with sprout ability will be able to take advantage of this relief in competition and resource use to increase in abundance, leaving late arrivals with less resources. So this scenario works really well in the contexts where native species are abundant in the landscape. However, in anthropogenic landscapes, invasive species are more abundant. And our management, the management of invaded forests is commonly addressed by attempts to control or eradicate the focal nuisance species. And by reducing plant biomass via the management, those actions lead to a temporary increase in resource availability and to low competition levels. And in this context, it's likely that priority effect, so the order of the species arrival, will dictate the diversity and the species composition to these systems that have been recently disturbed by the management actions. So if native propagules are scarce in our landscape, in this anthropogenic landscape, chances are that some invasive species or all the same invasive species or other invasive species could rapidly reestablish in those areas. So in order to investigate those mechanisms, I have three specific questions that I want to address. The first one is, what factors of the native community determine resistance to plant invasions and forest understories? And to that, I will perform a meta-analysis. The second one is, are invasive species tapping into unused resources and gaining a competitive advantage? To which I will collect some observational data. And the third question, are priority effects the mechanism driving community assembly after invasive species removal? And in order to answer this question, I will use a field experimental work approach. So let's dive into the first question, in which I performed a meta-analysis. So here for the meta-analysis papers, they were searched in Web of Science until the year of 2019. And in order to have the data extracted, these papers needed to compare two levels of invasion in the same forests. So in other words, low and high invasion levels by understory shrubs, and collected associated biotic and abiotic information. So here, biotic resistance, meaning, for example, differences in native species composition or abundance that reflected differences in invasive performance. Or abiotic resistance was driving the low level of invasives. Here, meaning environmental conditions under which invasive species performance was diminished. Here's just an overview of the study sites from which we could extract the data until now. And most of the papers, they are concentrated in the Northern Hemisphere, mainly in the US, but we still have some data points that are dispersed across the globe. And for now I want to present some preliminary results about this meta-analysis. So this forest plot has the effect size with confidence intervals on the x-axis, with mechanism on the y-axis. Any confidence interval that crosses the 0 line in gray there would be not significant. And the values, to the values of the effect size, the more negative they get, the stronger is the effect of a particular mechanism in forest's resistance to shrub invasion. So here the preliminary analysis show that both biotic and abiotic resistance are significant mechanisms of resistance to invasion with no overlaps in confidence intervals. But biotic resistance, biotic features of the forest's ecosystems, like native species diversity, abundance, richness, they had a stronger effect in constraining the shrub invasion. And when this resistance was split, given disturbance, if disturbance was present, absent, or unknown, biotic resistance was particularly strong mechanism of resisting invasion in the absence of disturbance. And when these two mechanisms of resistance were divided into papers that had only a single invader, one single species, and multiple invasive species co-occurring at the same site, biotic resistance was stronger under multiple invasive species invasion. So some next steps, I plan to finish extracting the data from the remaining papers, analyze the effect sizes as a function of other features like community type, and to access the mechanisms of resistance in more detail. So moving to the next two questions, they have some study sites that are in common, so I'll show them together, and then I'll dive into each of those questions. So the properties that I've collected data, or I will collect some data, are George Reserve, Stinchfield Woods, Mathai Botanical Gardens, Redrick Forest, Saginaw Forest, and Nichols Arboretum. And the order that I mentioned them is because they are in a gradient of urbanization from the less to the more urbanized setting as they get near to Ann Arbor downtown. And some comparisons of the abiotic conditions across 4 of those sites. Regarding to soil moisture, July was a drier month than June, and this trend happened regardless of site. With respect to light, in the bottom left graph, Mathai Botanical Gardens is a more open site compared to the others, and the total of nitrogen in the soils was lower in the botanical gardens compared to and the other sites were comparable. So here are some of the main invasive species. Some of those species I find in all of the sites, the 4 sites that I surveyed. And another species that is not here is the glossy buckthorn, that it's abundant at the botanical gardens too. So coming back to my questions, now I will show a little bit of the approach for the observational work. So 10 transects of 15 meters each, placed in a gradient of invasion from invaded to non-invaded areas within each forest stand, will be established. A pin will be placed in every 1 meter of the transect, so I have 50 points to sample within each transect, and I will collect data next year for this project from May to April of 2021. Soil moisture and light availability will be collected or estimated in every point within transect. And soil nutrient availability will be estimated using resin capsules at the stand level. And species composition and abundance of co-occurring native and non-native species will be collected within the transects. The selected traits that I will link to those species will be downloaded from TRI database and GROOT database. And only the missing values, the missing trait values will be collected in the field. And to try to estimate some intraspecific variability, I will probably collect leaf mass per area for each of those species that I surveyed. So I have a sense of how they vary within species. As I don't have data, I won't present the preliminary data for this one. So let's go to the next question and the final one about the priority effects driving invasion post-management actions. So I'm using a long-term experimental approach that I started to implement last year and I go into like 2022. So I have 3 plots grouped into 3 to 4 forest stands in each site with a total of 36 plots. Paired plots were set in invaded and non-invaded areas. Each plot has each invaded plot has 12 meters square of areas subdivided in small subplots of 1 meter square, and the non-invaded plot there is smaller, just 2 meter squares each. And treatments were applied in each of those subplots. So the first action was to remove invasive species from all of the plots, and this was done by the end of the growing season last year. And then some treatments were applied in each of those subplots. So one treatment applied is the repeated clipping, as single clipping is a common management approach, but can repeated clipping be a more effective approach to overcome resprout of the invasive species? So invasives have been clipped at the soil level in half of the invaded plots monthly during the growing season, and I started this repeated clipping treatment this June of 2020. Another common issue is the presence of soil legacy effects in previous invaded sites, especially with compounds in the soil that prevents native species establishment. And it seems that the addition of carbon have shown some promising results in other systems to overcome this issue. So in half of the plots, I applied sterilized straw. And in order to give the native species an advantage, so a priority effect, native species were seeded in the plots after invasive removal. And this treatment was applied to 5 subplots, being 1 non-invaded, and the others were the invaded ones. And I used 1 seed mix were just forbs, and another one were forbs with grasses, all of them native. And this treatment was applied by the end of this year, like October of 2020, so I won't show any data on this. So for the next few slides that I have shown, that I will show some of the preliminary data, the configuration will be the same. So I have a question at the top that I want to answer. Then I have the effect size and how I calculated that. So it's using native cover of the treatment subplot minus the native cover of the control of that subplot, control of the treatment at the subplot level divided by the average. And then I have each data point here is the effect size calculated for a plot by comparing to the 2 subplots. They will be shown here with the same graphs that I had before. And the graphs had the effect size on the y-axis, and they are plotted as a function of the environmental features, so light availability, soil moisture, and nitrogen. So to interpret the effect size values, positive values of effect size that here are in at the blue part of the graphs, they promote native species cover. The treatment promotes the native species cover. The effect sizes that are in the 0 line, the treatment had no effect on the native cover. And finally, the negative values of effect size are showing that the control instead of the treatment had promoted native cover. So going to the first question, is repeated clipping of invasive species necessary to promote natural regeneration or recovery? So we compared repeated clipping to single clipping subplots in natural regeneration plots, those ones that are highlighted to the left. And here, single clipping promotes regeneration, promotes native cover, with the effect size lines are above the 0. But the slopes there are not significant, so we can't draw any conclusions on the effects of the abiotic features. Also, this applies to all the other fitted lines, and the slopes, they are not significant, okay? And is this repeated clipping changed the effect if we are thinking about the seeded establishment? So is repeated clipping of invasive species necessary to promote the seeded natives? So it seems that here, repeated clipping has a positive effect on the native species cover when compared to the single clipping. So for simplicity, the next questions I will only answer using the repeated clipping treatment. So can priority effects be used as a tool to promote native species establishment? And we compared seeded and non-seeded subplots, and seeding tends to increase native species cover post-invasive species removal, as the effect size is generally positive. Do we have any soil legacy effects in the invaded plots? So invaded plots had a negative soil legacy effect on native species cover, especially under low light availability, soil moisture, and high nitrogen levels. And now that we know that we have a negative soil legacy effect, can it be amended by the straw treatment? So in seeded plots, it seems that straw had a negative effect on native cover instead of a positive that would be the expected result. But the straw seems to have a positive effect on the native cover even in the natural regeneration plots. So not in the seeded ones, but in their natural regeneration subplots. So to quickly summarize and wrap everything up, regarding my first question on what determined forest's resistance, the preliminary results shows that meta-analysis, in the meta-analysis shows that the biotic resistance is an important mechanism of resistance in forests, mainly in the absence of disturbance. My second question, I still need to collect some data, so we don't know that yet. And to my third question, investigating if priority effects are driving community assembly after invasive species removal, the preliminary results seems to agree with that, but there is a lot of variability on the data that needs further exploration and analysis. So with that, thank you so much, and I will take any questions.
Okay, thank you, Laís. And any questions? Okay, a question from Robin. Very nice talk, Laís. Did you find differences among sites given that they were subject to such different urbanization?
Thank you, Robin. Yeah, so when I analyzed those effect sizes in the last question about the priority effects, they do have different slopes. So I need to do further analysis to investigate on how different those slopes are in regard to each of those sites that we have for the project. I didn't do any further analysis, but I do know that they are different. They are different across sites. And I don't know yet if that difference is due to the urbanization gradient.
Okay, a question from Monique. So thanks, Laís, a very interesting and clear presentation. Any idea what the straw treatment is doing to the establishment of native species? Why would it have a negative effect?
Thank you, Monique. Yeah, I think that what's going on with the native seeded, like that when seeding natives to the plots, is that the straw might be covering them too much for the same. I think there are two things. Let me back up a little bit. The first one is that the straw might be for the first year. I seeded those plots last year, so then it's just like the first growing season of them. So they straw in the beginning or had a negative effect by covering them too much and slowing down their germination a little bit. It could be that, but I don't know. And the other thing is that the native seeds, the mix of native seeds, grasses or just forbs, they take some time to actually establish and grow in cover. So it's expected, that's why it's also like a long-term experiment. It might take 2 or 3 years until the native seeds that I seeded are actually established and bigger and with more abundance with higher cover. So that could be like a combination of effects. I would say that would be mostly because the native species take longer to establish as seeds.
Okay.
Yeah.
Okay. Second question from Paul. What aspect of reproduction does priority effects typically come from? Different seed germinalization rate, different rate of maturing, different seed dispersal ability?
Yeah, so here the priority effects, the way that I framed my explanation was more thinking about those species in which they have wind dispersed seeds, in those recently cleared areas, the little seeds, the small seeds wind dispersed, they would arrive to that area and fastly grow and increase in abundance. So like those species that are able to produce a lot of seeds, small seeds that would disperse fast and more effectively to those sites. I hope it answers your question.
Okay. So question from Krista. So I'm interested if during the meta-analysis there were differences in the biotic and abiotic responses based on different disturbance types.
Oh, that's a good question. So those are like we could figure it out that the presence and the like if disturbance is present or not would increase the resistance by a particular mechanism, so biotic resistance. But those, like knowing which type of disturbances are causing it is the next step. So this is a very interesting question. I don't have an answer yet, but I'm looking forward to know if there are any differences in which disturbances are there. Thank you, Krista. That was a really good question. Yeah.
Okay, question from Karen. How might you expect the effects that you are seeing so far to change in future years?
Oh, thanks, Karen. That's a really good question. So my hope is that the native seeds will be able in the next year and hopefully the next 2 years to establish better and grow and cover, and then we can have a sense on if they are actually promoting the periodic effects, if that would give them an advantage. And it seems that if they can establish, with the repeated clipping, I'm hoping that this treatment will actually work, because the invasive species in the subplots that I did not keep clipping the invasives are really huge again. They are not the same as it was when I removed them in 2019, but they're really big. So the native species, they don't have a lot of leverage to increase in abundance, but with the repeated clipping, as they take longer to establish and to grow, I'm hoping that they would have a higher effect, and so the effects for them would be more pronounced than compared to the ones with the single clipping.
Okay, other questions? Okay, if there are no more questions, I think we can go back to the questions for Poe. I think it's from Tierney. So how would you expect the diet breath to be affected in the migrating insects? Monarch, for example, are active for long periods of time, but in a specific area for shorter period of time.
Yeah, migration is complicated. That's a very good question. I would guess it would depend a lot where the where the consumption of resources was more directly related to the ability to reproduce successfully. Like you can find, I'm gonna go back to bees cheaply because I know them better, but I'll get away from them soon. If you know, if you have bees foraging and you just kind of like have these observation networks, you can say, hey, that bee landed on some particular flower. It must gather resources from there, but it may just be an adult getting a little bit of nectar and not actually producing a pollen basket. So not actually pollinating it, you know, so it's getting different resources. There may be situations in migrating animals where the adult has kind of a potentially broader range of resource availability than a juvenile or any kind of larva that needs to be procured for more specifically. And monarchs are interesting too because their larvae are so specifically focused in their diet, such that we can't really talk about diet breadth in that aspect of their of their development. But yeah, just to stop myself from rambling, I would say it probably depends on where the ability to get a you know, a reproductive unit up into adulthood and reproducing itself, like where that crux is probably going to be where the effect size is going to be larger.
Okay, so another question for Paul from Caleb. Do you think creating pollinator corridor across a region would affect these interactions?
I mean, like, yeah. So the pollinator corridors are interesting to consider. There are places, especially in Europe, I think the UK is doing a lot of this, where that's being floated as, or actually not even just floated, but implemented as a conservation technique, which is great. But it'll be interesting to see what use those have, depending on what flower species are planted. Yeah, if you know, kind of take away the natural fauna in an area and replace it with a bunch of flowers that have a different phenology, I would imagine that would have some effect on the realized foraging of the bees there. You may come into contact with physiological limitations, but yeah, if a pollinator corridor mimics the wild plant community, and they're kind of almost as a requirement mimics some of the native phenological patterns, I would imagine you'd be able to not have as much of a disturbance. But if you know, kind of just plant a flower because it's a flower without thinking about these other things, it's interesting just to think what might change. It may lead to competitive effects, such that something that's there for a long time had been picked up or had been kind of picked over before, whereas like a short-lived flower would spike up and then be foraged on by that short-living bee that happened to coincide with it. But if you only plant long-living flowers, then those resources may be taken before the short-living bee or the short flight period bee becomes active. So it's a fascinating question and kind of interesting aspect of conservation to consider that. Yeah, I guess I don't hear people talking about as much. So it's a good idea.
Okay, any other questions? Okay, so I think if there are no more questions, we can thank Paul and Laís again for giving great presentations today. And also thanks everyone for joining. And I'll see you in our next EEB seminar.
Thank you for the opportunity. Thank you.